Part 4 of the Biochem Study Guide. Hemoglobin also transports H plus and CO2. Hemoglobin also transports H plus and CO2 from tissues to the lungs and kidneys. CO2 produced by oxidation of organic fuels in mitochondria is hydrated to form bicarbonate. This reaction is catalyzed by carbonic anhydrase, which is abundant in erythrocytes. The hydration of CO2 shifts the equilibrium, causing an increase in the concentration of H+, which decreases pH. This decrease in blood pH, normally 7.4, influences O2 binding to hemoglobin. The binding of H+ and CO2 to hemoglobin is inversely related to the binding of O2. Peripheral tissues have a relatively low pH, such that O2 binding affinity decreases, and Hb, in hemoglobin, releases O2 and picks up H+ and CO2. In the capillaries of the lungs, CO2 is released, causing blood pH to rise, further causing O2 binding affinity to rise. The effective pH and CO2 concentration on the binding and release of O2 is called the Bohr effect. The binding equilibrium for hemoglobin and O2 is in the reaction shown, where O2 saturation curve of hemoglobin is influenced by the concentration of H+. O2 and H+ are both bound by hemoglobin, but with inverse affinity. When O2 concentration is high, as in the lungs, hemoglobin binds O2 and releases H+. When H+ concentration is high, as in the peripheral tissue, Hb, hemoglobin binds H+ and releases O2. O2 binds to the Fe2+ of the heme, whereas H+ can bind to different amino acid residues. In the peripheral tissues, when H+ binds to histidine HC3, it forms a salt bridge with aspartate Fg1 that helps stabilize T-state deoxyhemoglobin, giving the residue an abnormally high pKa in the T-state, whereas the R-state, which lacks this salt bridge, has a pKa of 6.0. As the concentration of H+ increases, histidine HC3 is protonated, causing a transition to T-state hemoglobin, which releases O2, because T-state is stable without O2. This also occurs when amino-terminal residues of the alpha subunits are protonated. So the four subunits of hemoglobin could communicate with each other not only about O2 binding, but also about H+ binding. They also communicate about CO2 binding, which is also inversely related to O2 binding. CO2 binds to the amino group at the amino terminal of each subunit, in the reaction shown. This reaction produces H+, contributing to the Bohr effect. CO2 binding also forms salt bridges, stabilizing the T-state and helping hemoglobin release O2. The concentration of CO2 in peripheral tissues is high, so some of it binds to hemoglobin, causing hemoglobin to release O2. Conversely, the concentration of O2 in the lungs is high, so some of it binds to hemoglobin, causing hemoglobin to release CO2. Oxygen binding to hemoglobin is regulated by 2,3-biphosphoglycerate. 2,3-biphosphoglycerate, BPG, is a heterotropic allosteric modulator, which is highly concentrated in erythrocytes. BPG reduces the O2 affinity of hemoglobin. BPG binding and O2 binding are also inversely related. BPG binds at a site distant from the O2 binding site and regulates O2 binding affinity of hemoglobin in relation to the partial pressure of oxygen in the lungs. BPG helps physiological adaptation to lower partial pressure of oxygen at high altitudes. When a person is moved from low altitude to high altitude, the concentration of BPG increases. This increase does not help hemoglobin bind to O2 in the lungs, but it does help displace oxygen from hemoglobin in peripheral tissues, thus promoting the release of O2. BPG binds in the cavity between beta subunits in the T-state, held there by interactions between the negatively charged groups of BPG and the positively charged amino acid residues. BPG thus stabilizes the T-state, which prefers deoxyhemoglobin to hemoglobin. If the T-state transitions to the R-state, the cavity is nearly closed, preventing BPG binding. Because a fetus must extract O2 from its mother's blood, fetal hemoglobin synthesizes gamma subunits rather than beta subunits, forming alpha-2 gamma-2 hemoglobin, which has a lower BPG affinity and thus a higher O2 affinity. Sickle cell anemia is a molecular disease of hemoglobin. A hemoglobin variation is the product of a variant gene, called an allele. Because humans generally have 2 copies of each gene, an individual may have 2 copies of one allele, homozygous, or 1 copy of 2 different alleles, heterozygous. Sickle cell anemia occurs in individuals who inherit the allele for sickle cell hemoglobin from both parents, fewer erythrocytes, and they're irregular shaped. As blood is deoxygenated, sickle cells increase in number, and the hemoglobin of sickle cells becomes insoluble and forms fibers. Sickle cell hemoglobin results from 1 amino acid substitution, a valine 6 instead of a glutamate 6, and the 2 beta subunits. The chargeless valine 6 residue creates a hydrophobic contact point at position 6 of the beta subunit on the outer surface of a molecule, and these hydrophobic contact points stick or clump together, thus forming fibrous aggregates. Sickle cells rupture easily, so people with sickle cell anemia have half the normal hemoglobin content. When heterozygous, the allele confers a slight resistance to lethal forms of malaria, but the individual should avoid stressing their circulatory system. Complementary interactions between proteins and ligands, the immune system and immunoglobulins. An immune response is an intricate and coordinated set of interactions among many classes of proteins, molecules, and cell types. An immune response distinguishes self from non-self. The immune response includes a specialized array of cells and proteins. 2 kinds of leukocytes, white blood cells, are macrophages and lymphocytes, and all leukocytes develop in the bone marrow. Leukocytes can leave the bloodstream and enter the tissues. Leukocytes produce proteins that recognize and bind to different molecules that may signal an infection. The immune system can be divided into the humoral immune system and the cellular immune system. The humoral immune system targets substances in extracellular body fluids. The cellular immune system destroys infected host cells, as well as some foreign substances. Antibodies, also called immunoglobulins, Ig, are soluble proteins produced by B lymphocytes, B cells, which complete their development in bone marrow, that bind irreversibly to foreign substances, targeting them for destruction. Antibodies comprise 20% of blood protein. The cellular immune response uses a kind of T lymphocytes, T cells, which complete their development in the thymus, called TC cells, cytotoxic, aka killer T cells. TC cells have T cell receptors, which allow them to recognize infected cells. Helper T cells, TH cells, are of the humoral immune system, and they produce cytokines, which are soluble signaling proteins, including interleukins, and interact with macrophages. In order to destroy pathogens, they stimulate the proliferation of certain T, C, and B cells that can bind to the particular antigen of that pathogen. This process, clonal selection, increases the number of immune system cells that can respond to a particular antigen. Every recognition protein binds to a distinct chemical structure. Humans can produce over 10 to the 8th different antibodies. An antigen is any molecular pathogen that can elicit an immune response, and it can be bound by 1 or more antibodies. The distinct chemical structure of the antigen to which the antibody binds is an epitope, or antigenic determinant. Haptens are small molecules covalently attached to large proteins, and they elicit an immune response. Antibodies are produced by this protein-hapten bond. Antibodies have 2 identical antigen binding sites. Immunoglobulin G, IgG, is the major class of antibodies, and it is in the blood serum. It has 4 polypeptide chains, 2 light, 2 heavy, joined by non-covalent and disulfide bonds. It forms a Y shape. There are 2 antigen binding sites between 2 variable domains, 1 light, 1 heavy. The FAB region, fragment antigen binding, and FC region, fragment crystallizable, can be separated using papain from pineapple. The FC region contains a bound carbohydrate. Bodies have constant domains called immunoglobulin folds, also known as beta sandwiches. There are 3 beta sandwiches in each heavy chain and 1 in each light chain. The variable domains form the antigen binding site. The overall structures of IgD and IgE are similar to IgG, whereas IgM is either monomeric and membrane-bound or a cross-linked pentamer, with 5 Ys attached by a J chain, and IgA can be a monomer, dimer, or trimer. IgM is made by B lymphocytes and is the major antibody in the primary immune response. Some B cells produce IgD with the same antigen binding site as IgM, but the function of IgD is unclear. Memory B cells initiate the secondary immune response. IgG is the major antibody in this secondary response. IgG is the most abundant antibody in the blood, and it binds to a pathogen, activating certain leukocytes such as macrophages. The macrophage then phagocytizes the pathogen. Receptors on the macrophage surface bind the Fc region of IgG, and when they bind to the antibody pathogen complex, the macrophage engulfs the complex. IgE interacts with basophils in the blood and with mast cells which secrete histamine in the tissues. The Fc of IgE binds to the Fc receptors on the basophils or mast cells, letting IgE serve as a receptor for an antigen, which, when bound, causes the cells to secrete histamine, causing the blood vessels to dilate and increasing their permeability. These 2 effects facilitate the movement of immune system cells and proteins to sites of inflammation. End of study guide.